welcome to the class. So today we will start off with learning more about the morphology of flowering plants. In our last class, we learnt about the flower, the different parts of a flower, androecium, gynoecium, and then we try to classify them: epiphyllus, epipetalus, ovary, placenta, placentation, estivation, and all those things. So today we are going to a much simpler, simpler structures, but very important structures in plants, and that would be the study of the fruits and the seeds. So we will start off to understand what exactly is a fruit. Okay, now we know that there was this flower, there was this ovary, and then inside we have ovule. All these parts we have already seen, right? So we have ovary, ovule, uh, and all this. Now this uh, ovary is what we call as the fruit. Okay, ovary becomes the fruit, very important one. And you know what exactly ovary is? Ovary is that the gynoecium, the outermost covering of this. Now this is what is called as ovary, fruit. Sorry. Now we have a fruit, right? Now this ovary develops into fruit. What makes the ovary to become a fruit? Obviously, it is the ripening of the ovary or the maturation of the ovary which makes it into a fruit. And what makes it, what gives that characteristic of, modifying that ovary itself into a fruit? Now that is dependent on the process of fertilization. Now during fertilization, we have seen that the pollen grains would go inside the ovary, enter into the ovule, and there that would get fertilized. Right? This is what would happen. Now after fertilization, there would be some morphological changes in the ovary, and these morphological changes will finally help in the formation of the fruit. Okay. Now this kind of fruit is something that we generally see. Okay, this is the how the fruits are generally formed. But however, sometimes, very rarely, you see that even without fertilization, the ovary undergoes the modifications to form a fruit, and such fruits are what we call as parthenocarpous fruits. We call them as the parthenocarpous fruits. So what exactly do we mean by parthenocarpous fruits? Here the fruits are formed because of the ovary undergoing modifications. But the modifications is not triggered by fertilization, but they are triggered because of the age of the plant, or it might be because of certain chemical changes happening inside the gynoecium. Now they bring about the formation of parthenocarpous fruits. Always remember, if you have a fruit and you need to find out if it is a parthenocarpous fruit or a normal fruit, it is very easy to find out. See if it has seed. If it has a seed, then always it is formed because of the fertilization, and that would give rise to a normal fruit. If there is no seed, then that would be a parthenocarpous fruit. I am sure all of us would have tasted this seedless grapes. Now this seedless grapes are seedless because they are all parthenocarpous fruits. They are all parthenocarpous fruits. So that way we see the ovary forming the fruit. The ovary forms the fruit. The ovary forms the fruit. Now the next thing that we should see is what is the structure of this fruit. Now I'm sure all of us would have seen many types of fruits. Take grapes, take mangoes, take pomegranate. You take any fruit. Generally, you see that there is this stalk, and then you would have the most. So you would have a mango. We will take the example of mango. You can take any fruit for that matter, but since your textbook says mango, I have taken a mango here. You see that there is a very, very thick outer wall, right? Outer wall, which will be green in color but later turns yellow. And then inside this, you see a pulp, right? You see a pulp, and in between, you see that there is a seed. This is how normally a mango looks like. Now, always this is outer membrane is what we call as epicarp. Remember why we get the name as carp. Carp is actually for the carpels, the number of divisions that we see in the gynoecium, right? Carpels. So this carp basically means that epicarp, it is covering the entire surface. Okay, so basically you can say a segment or a membrane. So epicarp is basically a membrane which is there on the outer surface. Okay, then you have the middle one, which is called as the mesocarp. Then we have the middle layer, which is called as the mesocarp, and then inside this we have something that we call as the endocarp. So we have the epicarp, the mesocarp, and the endocarp that is present inside. Now inside this endocarp, what you see is actually our seed. It is our seed, the mango seed. Now always epicarp is something that is kind of thick. You can see that it changes its color as and when it starts aging. You call that as the epicarp. Then in between, we most of the time see a thick pulpy layer of the mesocarp. Now mesocarp is most of the times rich in sugary substances, and this helps in uptake of uh, nutrients, gives it to the seeds, developing seeds, protects the seed also, has good amount of water content. And then we have the endocarp, which is again kind of hard or soft depending on the fruit, and again protective in nature. So these are the three layers of the fruit: the epicarp, the mesocarp, and the endocarp. Now suppose you have a pomegranate, you will see the similar uh, outer layer, middle layer, inner layer, and then inside you will see seeds. So depending on what kind of flower it is, you will see the presence of so many seeds present. Okay, in mango it is a monocarpellary flower, therefore you will see one seed like this, right? So depending on the flower, you see the seeds very differently. Suppose you have the pomegranate, the flower is very different, so you can see the arrangement of the flower also very differently. So epicarp, mesocarp, and endocarp, all these are the different parts of the fruit. Now this fruits that we are talking about are group of fruits which are called as drupe. What is the difference between fruit and drupe? Drupe is just type of fruit. Okay. When we are talking about only fruits, there are very different varieties of fruits. Some of them, for example, if you take pineapple, can you consider the pineapple structure to this? It is slightly different. How about a banana? Banana structure is entirely different. So based on the structure of these fruits, we have different varieties, and drupe is one of that kind of uh, fruit. This is basically, a monocarpellary. You know why it is monocarpellary? Because it has only one major seed there, monocarpellary. Then it has the superior ovules. Okay, it has superior, not ovules, sorry, superior ovaries, and because of this, you see that they are one seeded. They are one seeded. So this is how the fruit is. Okay, so we call such kind of fruits as drupes. One more example for this. Is our coconut. Coconut is also considered to be a drupe. Okay, you can see that there is epicarp, mesocarp, endocarp. All these three, we see that mesocarp is kind of edible here in case of most of the fruits which are drupes. Okay, but there is a stony hard endocarp. Okay, there is a stony hard endocarp. So mesocarp can be edible or it cannot be edible, or it can be fibrous, so on and so forth. So there is good amount of diversity there again. But what makes a drupe is basically one large seed, monocarpellary, and coming from the superior ovary. I hope you remember what superior ovary means, right? The ovary is situated on the uh, top. So that would be our fruit, right? Now inside the fruit is the seed. So how are these seeds present? Now inside the fruit we see the seed. Now seed is something that is very, very important. Why? Because this is the one which has the ability to give rise to the young ones, right? Seed is something that is very, very important, because this is the one which can give rise to the young ones, new plantlets. So where is the seed coming from? We have seen that the ovary gives rise to fruit. Here we see that ovules gives rise to seed. Okay, the ovules gives rise to seed, and always this seed, since it carries the young plant, right, it has to be protected, and you always see that it is embedded inside a seed coat. It is embedded inside a seed coat. So that is about the seed coat. Now when we look about the seeds, there are basically two types of seeds that we can see. What are they? We call these seeds to be grouped as either monocotyledonous or monocot, monocotyledonous seed, and then we have the dicotyledonous seed. So we have monocotyledonous seed, and then we have the dicotyledonous seed. So what is monocotyledonous seed? Monocotyledonous seeds are those seeds which have some kind of structures. Okay, they have monocots. The cotyledons, there are only one cotyledon. Okay, so if they have a single monocotyledonous structure, single cotyledon, we call that as a monocotyledonous seed, and we call such seeds as monocots and the plants as monocot plants. Similarly, if we see two cotyledons, we call it as the dicotyledonous plants or dicotyledonous seeds. You cannot see more than two, so thankfully we have only monocots or the dicots. Okay. Now, how to find out if it is a monocot or if it is a dicot? Very, very simple. Take any seed and see if you can break it into two equal halves. If you can break them into two equal halves, it's a monocot. If you sorry, if you can break them into two equal halves, it becomes a dicot. If you can't break them into two equal halves, it is a monocot. So very simple to do that. Okay, so now let us see the structure of dicotyledonous seed. Very simple. So for this we will take our one of the pulses. Okay, kadle bija. We will take the example of the kadle bija and see how the kadle bija looks like. Okay, it looks more or less like this with a slight indentation here, right? If you see that, you see the slight indentation, and if you observe very carefully, you will generally see a small region which has come out like this. You would have seen this, right? Now this is what is very important for us to tell if it is a dicotyledonous seed. You can see that there are two equal halves which are joining together here, a very important feature. Okay, now this dicot seed is not present alone. Obviously it has to be present in the fruit, right? And we have already learnt about the placentation. So this seed is attached to one of the membranes there, the placentation there. Now this region to which it gets attached to the membrane is what we call as the hilum. We call it as the hilum. Okay. Now very close to hilum, you see that there is one small pore in this region, and that small pore is called as the micropyle. Okay, we call that region as the micropyle. It is called as the micropyle. Now I am sure you would have done this. You take this kadle bija and you put it in water, soak it in water. Okay, after a few hours, if you just take this from the micropyle, you can take out one brown colored peel, right? Now that brown colored peel is what we call as the seed coat. We call it as the seed coat. Now if you carefully observe the seed coat, there are two regions or two layers of this seed coat. Okay, so we have the outer one, which is called as the testa. Testa is the outermost coat, and then we have the inner one, which is called as the tegmen. So we have the two seed coats, testa and tegmen, that is present in the seed coat. Now what you can do is, you take out the seed coat, open the testa, tegmen, then you can also see the presence of hilum and micropyle. I want you people to see this. This is all, that is when you can kind of observe how beautifully this dicot seed is. Then you open it up like this. Okay, you open it up like this, and when you open it up, what you see is, you see that you can see two almost equally shaped, similar shaped parts of the seed. That yellow colored thing that you see in the case of kadle, now that is what we call as the cotyledon. That is what is called as the cotyledon. Okay, that is what is called as the cotyledon. Now if you just very carefully open it, you see that these two cannot get separated. They will be kind of closely held together, right? Now if you just look in between, you can see that there is some kind of structure like this. You can see a structure very much similar like this. This would not be this big, this long, it would be like this, right? So you would see a structure like this. Now this is what is the actual seed, and that actual seed is what we call as the embryo. That is what we call as the embryo. So we have one region which is able to give rise to the shoot parts. We call that as the plumule, which we have already learned, right? So we have the plumule, and then there would be one more region here which would be called as the radicle. So like this, we have the dicot, okay, dicot having the hilum, the micropyle, the testa, tegmen, and you open it, you have the plumule and the radicle. So this is how is the structure of our dicot seeds. I hope you got it right. Now when we go to monocots, that is completely different. Okay, that is some completely different. We will talk about this monocots now. When you take about monocots, you do not see this kind of two cotyledons present, but you have only one monocot, one cotyledon that is present. I am sure you would have noticed this jola, corn. Now, if you have noticed the corn, it looks more or less like the structure of our tooth, right? It would look more or less like the structure of our tooth. It would look more or less like our tooth, having one outer coat. Then there would be something like this, right? And in the middle, you would have seen this kind of structure like this. Like this. So the structure is kind of completely different compared to that and compared to this. Now what is the difference here? Now here we see that there is the embryo, right? Like how we had the embryo showing the radicle and the plumule here. Even here we have the embryo, but that embryo is this. Okay, we have a embryo which is this, and then covering the embryo we have this kind of small layer. Let that be there, and covering that you have one more layer here. Now can I call that as a cotyledon? Actually speaking, no, because this whole thing is what we call as the monocot or the cotyledon. Okay, now this outer covering, you see this here, has uh, a very important region. Okay, a very important region which we call as the endosperm. We call that region as endosperm. Now what is this endosperm? I will tell you more about this endosperm after I finish the entire uh, you know uh, seed structure. So we have this endosperm. As of now, just know that endosperm is basically uh, that part of the fruit which provides nutrition to the growing embryo. Okay, you just get to know that there is this endosperm. Now above this endosperm, there is one layer, right? Now that layer is what we call as the alurone layer. What do we call that as? We call that as the alurone layer. Now there is this alurone layer. Inside this alurone layer there is this very fleshy endosperm. I'll tell you why it is fleshy and what is its function in some time. Then inside that we have you know uh, one cotyledon. This whole thing that you see here, that which is called as the scutellum. We call that as the scutellum. So we see the presence of shield shaped scutellum, and inside the scutellum we have the embryo. And obviously our embryo has two regions, which is the radicle, and then we have the other one which is called as the plumule. So we have the radicle and the plumule. Right? Okay. Now, after we talk about the radicle and plumule, okay, if I by chance take this embryo and I try to break this open, I do not see a structure similar to this, but I see a structure slightly different. Okay. I'm going to write the structure of what I see in this region, or maybe I will erase this entire part and I will write it here. Okay. So I hope you understood what is there here. So you have the outermost layer, aleurone layer, inside endosperm, and then you see the scutellum, which is nothing but our cotyledon. Inside that we have the embryo. Now how does that embryo look like? Let us see that. If by chance this is my monocot seed, okay, we have the outermost covering here, the outermost covering, which is called as the seed coat, or we also call it as the fruit wall. Okay, the fruit wall or the seed coat. So you see that here, and inside what you see is that endosperm, right? So you see most of the endosperm that would be present here, covering almost 3/4 or uh, slightly lesser than 3/4. Okay, so much of the seed is covered by the endosperm, and we have already seen that the endosperm is having an outer layer which is called as the aleurone layer. It is called as the aleurone layer. So now we have the seed coat, just below that we have the aleurone layer, and then below that we have the endosperm. Now if by chance I dig deep inside here, then what I see is the membrane here, right, and that is what we call as the scutellum. That is what we call as the scutellum. Now inside the scutellum we see the embryo. And how is the embryo? Our embryo looks more or less like this. Okay, if you just take the embryo, you can see something like this, right? You can see a structure like this, and here you can see that there are some structures present. Okay, there are some structures that are present here. So many, but I will write. You would see structures like that. Alright, so this would give rise to the shoot part, we will call it as the plumule, and then this we call it as the radicle. Can you see this part which is kind of covering the entire embryo here? Now that would be our region of the embryo, right? Now this plumule is covered by a layer which is called as the coleoptile. We call that as the coleoptile, and then this lower region is what we call as coleorhiza. Rhiza for root. So we have coleoptile and the coleorhiza that is present here. So that this is how our monocot seeds would look like, right? Okay. Just to give you another quick review of what we have seen that in the case of dicot there are two cotyledons. Open it up, you see the plumule, and the radicle, and both of them present there, and you can very clearly make out what the structure is. In the case of monocots, you see a structure very much like our tooth. You see the outermost layer, the endosperm, then inside you see the embryo having a layer which is called as the scutellum, and inside that you have a region which gives rise to shoot, which gives rise to root, call it as the plumule, then you call it as the radicle, both of them covered by coleoptile and the coleorhiza. right? So this is what we actually look for in the case of our seeds. Now in seeds, we would like to talk about one particular part, and that part is what we call as the endosperm. We call as the endosperm. You know, endosperm is basically one region in the plant which actually provides good amount of nutrition to the growing seed, right? The growing seed does not have any kind of uh, circulation there, no transcription, no translation, nothing, right? They are basically the accumulation of the food products. Now all this is possible mainly because of the endosperm. Endosperm, most of the times we see that it is very much like, you can compare that to the amniotic fluid of uh, the seed. It is kind of giving all the nutrients that the seed requires, and finally you see that by the time the seed matures, the endosperm might be lost. And if the endosperm is lost, then uh, then nothing. The seed is completely matured. You take the seed and the seed would have all the nutrients which our endosperm had. Have you seen this pulses, the one which we just wrote now here? That does not have an endosperm. Did we mention about endosperm there? We did not. Why? That is because most of the dicot plants, in the process of development, the endosperm would be lost. The endosperm would be lost. But in the case of certain monocots, not certain, most of the monocots, you see that the endosperm is retained for quite a long time. If you have seen the jola, right, if you have seen the corn, you can still see, I want you people to take one corn, keep it towards the light, and you can see one yellow colored structure and then one middle white colored structure. Now that yellow colored structure is the endosperm, has all the good nutrition. Most of the monocots have this endosperm very prominent. In fact, the copra that you eat, coconut, right? That coconut is the drink that you have, coconut juice, standard coconut juice, that is also the endosperm. Okay, the jola that you eat is endosperm. Most of the endosperm that we talk about, all these are very nutritious. And why are they nutritious? Because they the growth of the support the growth of the seed. So that is the reason why we see that endosperm is quite important. Okay, most of the dicots would have lost it, but you know, in some of the dicot plants, like if you know castor, castor is that oil from the castor oil, right? The castor seed, if you take and you try to see that if you can get the castor seed, you can look into it, you see that the endosperm is quite prevalent even after the seed is quite mature. But in most of the monocots, you see that the endosperm is very common, very common. Only under conditions where this whole thing gets dried up, then the size of the endosperm shrinks. The size of the endosperm shrinks, and then you see the embryo, the scutellum that is present. But even then, you see one this very small thin layer of the endosperm that is present. Okay, so this is about the seed. This is about the endosperm and the fruit that we can talk of. I hope you don't have any doubts with this. So right now we are in a position to understand which part of the plant comes from where, right? Then we can also tell about the various diversities, the morphology of the root, the stem, the leaves, then the flower, the fruit, and the seeds. However, we have not spoken about different types of fruits and so on, but all that, you know, we can learn as and when we come across them, or maybe, you know, when we really would want to talk about them under specific conditions. But as of now, you should know what exactly is a fruit, what exactly is a seed, different important parts of the fruits and the seeds, right? So now we have seen about all these plants, and now what is left is to talk about three different families of the plant systems. Okay, we have seen about the plants, we have seen about various diversities that they can exist there. Now what we should know is about three families: Fabaceae, Solanaceae and Liliaceae. Okay, now these are the three families for which we should know how is the plant, what are their characteristics, what are their fruits, what are their flowers, and so on. Okay, now this is such a nice part of botany. Frankly speaking, this is the real botany, because this is something which is very interesting, provided we do some activities here. Okay, now I would like to do this part of biology, this part of understanding the families in a completely different way. What I want you people to do is, when I am doing this, I want you people to have these plants in front of you. Now that you might feel that it is not so easy, but the plants that I am going to tell you is not something that is very difficult for you people to get. Okay, you can get can you get moong dal, or any dal for that matter, any pulses that we see. Okay, moong dal or kadle kalu, any dal, if you can get one of that kind. Then if you can get a potato or a tomato or a chili plant, and then if you can get one tulip plant or asparagus, that would be really great. Okay, otherwise I can show you with pictures, but that would not be that interesting. Okay, so I would I would want you people to get that plant with you, and then I will also get that, and when I am dissecting, we can actually do it very well. Okay, so that is one part of our understanding the morphology of flowering plants for three families, which I will tell you as and when we are dissecting the plant. Okay, so this is about this chapter. In our next class, we will start off with the anatomy of the flowering plants. But I will, this part of classification will be included as and when you tell me when you have got the plants, and as and when I get the plants, and we will do it as a activity. Fine. Okay then. So if you have any doubts, make a note of it. We will discuss in our next class. Okay.